Good morning, friends. Continuing the yesterday topic, we studied previously transfer RNA, transcription unit, and central dogma. And today we will slightly study about the RNA. Because RNA is made of single polynucleotide chain. This is phosphates molecule. This is ribose, and in DNA deoxyribose there are nitrogen bases adenine, guanine, thymine. No cytosine in place of cytosine. It has uracil. RNA is of three types. Sorry, you studied messenger RNA, transfer RNA, and ribosomal RNA. You have studied about transfer RNA yesterday. Friends, this is a beautiful diagram of DNA. I usually make, first of all, guanine combined with cytosine with double covalent bond, adenine combined with guanine combined with cytosine triple covalent bond, and adenine thymine combined with double covalent bond. This is deoxyribose sugar, and these are the phosphate molecules. Second, the two bases unite by hydrogen bonds or covalent bond. The base and the pentose sugar combine with that very glycosidic bonds, and the pentose sugar and phosphate combine with phosphodigester bonds. Then this is anti-parallel direction. One polynucleotide chain is in 5-3 direction, and other polynucleotide is 3-5 direction. In layman language, at one end the oxygen is carrying in one direction, at other end oxygen is carried pointing in opposite direction. This is called anti-parallel direction. This is triple phosphate bonds making adenosine triphosphate. This is adenosine 3-phosphate triphosphate, and ATP loses one phosphate, gives energy, adenosine diphosphate, and if one phosphate, adenosine monophosphate. So one unit of DNA is nucleotide. Nucleotide is made of one base, one sugar, and one phosphate. And without phosphate, it is called nucleoside. Base and sugar is nucleoside, base, sugar, and phosphate is nucleotide, and long chain is polynucleotide. Normally it is coiled structure, helix, like the staircase of the helicopter, rope staircase. The ropes are polynucleotides, and the horizontal rods are the base pairs. Friends, next question is ribosomal RNA. You must have not seen the diagram of the ribosomal RNA. This is the ribosome, smaller subunit and bigger subunit. This is 70S ribosomes in eukaryote and 80S ribosomes in prokaryote. S is Svedberg unit, measuring unit. So this ribosome is made of ribosomal RNA and proteins. Ribosomal RNA and proteins. We will study this diagram, a polypeptide chain formation in protein synthesis. Union of amino acid is helped by ribosomal RNA. My dear friends, our next beautiful question of today is transcription. The meaning of transcription is formation of messenger RNA from DNA. DNA shows replication, that means duplication of DNA. The steps you studied in replication of DNA were slightly different than transcription. Similarly, as in replication, DNA opens like a zipper fashion. In transcription or making of messenger RNA is same process at the point where the sequence of the bases is formed that make a bubble, or at that point the coiling of the DNA is uncoiled. uncoiled. The DNA gives information to the messenger RNA to move to ribosome to make protein synthesis. Because messenger RNA, transfer RNA, ribosomal RNA helps in protein synthesis, and formation of the RNAs is must in the nucleus, and they are given out from the pores. The formation of the RNA makes opening of the that very DNA, that and DNA template is formed. Two DNA templates are formed, and complementary strand is formed in replication. But in transcription, only single template is used to make messenger RNA. Not two DNA templates are used, because RNA is separated and DNA is reformed. First there is uncoiling, then there is recoiling. It takes place in interphase. So RNA polymerase 1, 2, 3 helps to bind the promoter site of the point of the template